Okay, so it's 1 minute past the time. We will not have a full house, but this is being recorded, and because Ewan never says a word that is not interesting, I encourage you to see the video, even if you have been here, right? So, as a way of introduction, I should start from personal experience and move to the most serious stuff. And I remember, sorry, this we didn't prepare, and perhaps he doesn't like it, so who knows. So let's start with the less important and the most important stuff, because of course a presentation of Ewan could be as long as his talk, right? But we are not going to do that. So I do remember, as personal experience, going to Cold Spring Harbor meetings in the early 2000s and zeroing in a guy who would never sit down. He would always stand up and ask questions every talk, right? Then, after one meeting, I learned that this was the only sensible way to be awake with the jet lag. You stand up, so you are not going to fall asleep, right? So that's a trick I learned from him. But more seriously, Ewan is a voracious biologist, and I would say a voracious Twitter person as well. He knows a lot about biology, and you can learn next to him many different things of many aspects, both theoretical, empirical, and practical. I mean, he has done beautiful things. For instance, he's the first person I am aware of that actually had his group store information within the DNA of some cell cultures, right? That's so very cool, and many other things like that. Because also, on top of being a scientist, a voracious scientist, he's apparently a fairly sensible administrator. He has all these, they have disappeared from here. He has all these titles. He's in charge of the European Bioinformatics Institute together with Rolf Absweiler, I don't know how to pronounce it. He's the deputy director of the MBL, and he occupies several different positions in several different institutions and companies, right? He's the chair of the Global Alliance for Genomics and Health, which is a big worldwide initiative, very fundamental. He also is one of the directors of Genome England and so on and so forth, whatnot, right? So this is already a lot of things, and I could go on. But the most important stuff for you who are mostly from Barcelona and Spain is that he's the person that you will ever meet that is more similar to, I don't know, Sir Galahad or Harry Potter. Because, you know, he's a commander of the British Empire, so we all hope that eventually we will have to call him Sir Ewan Birney. Well, we'll have to wait, but it will get there. And also, he's Harry Potter because, you know, he went to a boarding school in Eton and he had all these houses and things. So let's hope he does a bit of magic for us. Thanks very much, Ewan.
Yeah, thank you. So I hope this will be interesting. It touches on a number of different topics from EBI and broader EMBL. So just a quick introduction to EMBL for the people who don't know EMBL. So it's the European Molecular Biology Laboratory. We're headquartered in Heidelberg, Germany. And we're kind of a child of the 60s or the 70s. At that time, Europe did federal European research by setting up these treaties which countries joined. Now, probably the most famous of those is CERN. I hope that soon the second most famous will be EMBL. And the European Space Agency, European Space Southern Observatory are other places. When it was founded in Heidelberg, Germany, we basically have two missions, which I'll come on to. One is doing research, but the other is delivering services. And at its point of foundation in 1974, it had two outstations, or two other place sites, next to these high-energy physics machines, synchrotrons, to provide access to those synchrotrons. And then later on, in the early 90s, a fourth site was added, which was EMBL-EBI, just south of Cambridge in a little village called Hingston, and it's on the same campus as the Sanger Institute. Then after that, Rome was added doing epigenetics and neurobiology, and then the most recent addition, which I'll be going to this afternoon is EMBL Spain, EMBL Barcelona, in the lovely PRRB building just next door. And I know James and the people there have a wonderful time doing tissue biology and organoid disease modeling. So EMBL was founded with these 5 missions, and the first 2 of those 5 missions are like the core mission. So one is to do excellent research, to be a place which enables often very young scientists to grow and then go back to wherever they want to go to afterwards. And we've been a bit of a beacon for many other institutes across Europe, and I hope Roderick would agree that it was part of the inspiration to the CRG when the CRG was built to have this young turnover model, or founded. But as I mentioned, the other aspect of this is delivering scientific services, and that is to enable science to happen, not to do science, but to enable science to happen, to enable scientists to get access to synchrotrons, very, very coherent and bright X-rays, often used in X-ray crystallography, and to get access to data. And that is what the unique thing that EBI does, is provide access to data from around the world to enable science to happen. We have a new Director General, Edith Hurd, and she has challenged us at EMBL and made us step a little bit further out from going from atoms and molecules. EMBL would often sort of end at this sort of organismal scale. But Edith wants to get us out of just studying life in situ or life in controlled settings and get out of the lab to also talk about how organisms interact as populations, in communities across many different organisms, and then finally in ecosystems. And this means that we have added a variety of new different components to EMBL's scientific scope, and in particular thinking about the entire planet, which is quite a bold thing, but all of life which is on it is part of that. And the other aspect is to think about humans, but humans in their ecosystem. And humans are interesting here for 2 reasons. The first reason is that we create an ecosystem that we carry around in us. That's our gut and our gut bacteria and these other things. So we have a very close relationship to a whole bunch of different organisms. And the second thing is that we create a lot of ecosystems by our behavior. And so we are very active behavioral animals, which affects each other and affects the world around us. We build buildings, we change things. And so those 2 aspects of human ecosystems is important to remember. So this, we hope, is going to be a new era of molecular biology at EMBL, we hope across Europe as well, where in some sense we're not only studying life because we want to understudy life in humans, and ultimately the reason why taxpayers fund us is often so that there can be health benefits, but also we want to study life to understand how life works across the planet, and in that sense help ecologists and environmentalists and save or help understand how we can improve the environment around us. So I just want to go back to EMBL EBI. So this is the institute which I direct along with Rolf Abweiler. We're the 2nd largest part of EMBL, and we're totally computational. We're about 750 people. It is all dry. We don't do experiments at EMBL EBI. We just manipulate data. And if you notice, I've swapped these 2 around because this first one is by far the most important one, which is we are the European part of global collaborations to coordinate data sets. And the most striking one 2 years ago was the reference human genome. So if you use the human genome, you either use it directly from us, or you use it via another resource, which has come. We are the master part of, we're the European part of the European collaboration, of the worldwide collaboration for the reference genome. It's not only the genome, but it's the annotation on the genome where the genes are. It's not only that annotation of where the genes are, it's their protein structures. It's not only their protein structures, it's how they form into pathways and how they turn into cells or drive cells and deliver or make one of us. And we don't only do that for humans, we also do it for mice, for platypus, for breadwheat, for plankton, and even for little viruses. And so you can imagine that when the pandemic struck, in fact, before the pandemic was totally announced, we already were repositioning some of our data resources to be ready to handle what we knew was going to happen, which was a very large-scale investigation of this virus. Now, I'm going to come back to the pandemic, but I just wanted to talk about a real success story that had happened, in fact, over the pandemic. And that is this incredible work by friends and colleagues in Deepmind, a company owned by Google, or owned by Alphabet, more accurately, who have led the charge of deep neural network learning, or artificial intelligence, if you like. Demis Hassabis, who heads the company, has an amazing ambition to be able to solve fundamental scientific problems using computers. And arguably, this is the first time a big fundamental scientific problem has been solved using this sort of technique. And this is this very simple problem, which is that proteins form, most proteins form precise three-dimensional globular shapes. And we know from experiments done in the 1970s that the only piece of information behind that is the amino acid sequence of the protein. If you put a stretched out piece of amino acids in the right buffer conditions, it will fold into a globular protein. So somehow the structure is determined by this sequence. Despite the fact that we know that this happens in real life, that it's a physical truth, it has been incredibly hard to take the information of just the amino acid sequence and create a three-dimensional structure. And that is the problem which they solved in Deepmind. Now, it's important to stress they didn't do quite the problem that I've stated. So what I've stated is you take one protein sequence, and Deepmind allowed themselves, as most other people who do protein folding do, to also use information from evolution. So you take a multiple alignment of proteins, which you believe are related by evolution, and you use that multiple alignment to predict the protein structure. Now, interestingly enough, it doesn't have to be that deep. You can do it with about 10 or 20 protein sequences. It's not that these have to be super deep structures. You just need enough for this AI algorithm to latch onto. And I'm not going to tell you about the paper is really worth a read. Demis Hassabis says that it is the most complicated piece of AI his team has generated. And one of the most important things is the casting of the problem. So how does the neural network see the problem? And it is not as XYZ coordinates. That's the most important thing to realize. So in the design of the AI system, they changed the problem representation. It's the same problem, but they changed the representation so that the AI system could use the multiple alignment and then learn how to do this. I should have probably put another slide in here, but this is John Molt, who runs the CASP competition. So it's worth another interesting fact here. Two interesting other facts here. So firstly, why was this problem solvable? So it was a well-described problem, it was well understood, but there was also 40, 50 years of deposition of public data into the worldwide PDB database. And so that 50 years worth of data was key to producing the data sets that they could train on. So that's number one. And the second reason why they selected this problem as one of their sort of flagship problems to tackle was that there was a community who would assess the predictions. And this is what's spinning here is a prediction versus reality, and this is the CASP competition run by John Moult. He started this in the 1990s, and he started it because everybody was claiming that they had solved the protein folding problem, but as soon as after the paper had been published and other people ran the code, it never really worked. So in other words, they were either overtrained or just plain had fooled themselves into thinking that they had come up with a good prediction. And so John started a competition. The competition was very simple. He would get protein structures that his colleagues were going to experimentally solve. And then he would say to computational groups, you must give your prediction before it comes out publicly. And he was very careful about how the timings worked. So the people who make predictions have to make predictions without knowing the answer. And this revolutionized the assessment of protein structure prediction. It showed that everybody was pretty awful, but it meant for Demis that there was a way of assessing whether they were doing well, and that the way that the scientific community knew. And that's one of the reasons why why Demis chose that problem, and why this has been so successful in some sense, so well understood. Now, at EBI, we've done two things here. We have provided the protein data bank, UniProt, the protein sequences, and provided advice when asked, as we would do to anybody, about how those different resources work. And for a number of years, Deepmind has been talking to us about how do you precisely use this or something else. But the second thing they did is, I think very nobly, brilliantly, Deepmind decided to make all their code open, their methods open, their code open, and to make predictions by running AlphaFold over many, many proteins. And my colleague Samir Velenka at EBI, he took on the challenge for storing the results of the prediction. And that's called the AlphaFold database. It's run at the EBI. It's going to have, would you believe, 1 million protein structures sometime this year. And sometime next year, they expect to get it to 50 million or 100 million protein structures. Now, the reason why they can get such a large number of protein structures, of course, is because AlphaFold is a computer program. It's not an experimental technique, it's a computer program. And so they can generate all of these different structures and store them. Now, they're not perfect, but they are very good. And importantly here, the program has an internal scoring system about whether the program believes it's made an accurate prediction. And so it is a reasonably well-calibrated system. So in other words, when the program believes it's made an accurate prediction, you can normally trust it. When the program says, I'm making a prediction, but I don't believe that it's accurate, you can also trust that it hasn't made a good prediction at that point. And this just is in January, we released this along with Deepmind, 200,000 new structures from 27 different proteomes. And this is all these different neglected tropical diseases and bacteria in the data set. And I think this is where AlphaFold and other computational techniques will have a really big impact, because you can apply them to places which are less sexy, less interested, get less funding. So often people think about this as the benefits to understanding human biology. I think understanding these neglected tropical diseases is going to be far, far more important. Now I'm going to switch a little bit to SARS CoV-2 and what we did during the pandemic. And again, we pivoted all of our different areas to looking after the pandemic, responding to the pandemic. So our research, our scientific services, and training, we ran training courses. We partnered with many different technologies and companies, and integrating the life science is research across Europe, helping to integrate that. So I'm going to just touch on a number of them. I'm going to dive into a piece of research. So Hamburg's beamline is still used today as one part of the QA process, the quality assurance process of the BioNTech vaccine for the lipid nanoparticle. So they measure the lipid nanoparticle using the SARS beamline on Hamburg for a subset of the viruses that they produce. And we also had a large amount of protein structure work that was done in both Hamburg, Grenoble, and Heidelberg. We've done a bunch of different pieces of collaborative work, both computationally and in experimentally, about how the virus infects, the structural properties of the virus, and I'll come on to a piece of work about the infection patterns of the virus. And then we produced this entirely open COVID-19 data portal with the backing, in particular, of the EU, a really good, deep collaboration between EMBL and the EU to deliver very quickly, within a week of the pandemic being announced, a platform to allow people to openly share the SARS CoV-2 data, and in particular the raw sequence data, which has become very important in being able to track down subtle and complex errors in the genome sequence. This is just one of these, like we do a lot, slides, the COVID-19 data portal, so you can come and go there. So one is the viral sequences. We also have a place to coordinate controlled access to human sequences. This is in collaboration with the CRG here on the EGA work. Expressions of data sets associated with expressions, a lot of protein structures. There are some drug targets and chemical screens that were run that were stored, and we coordinated the literature, including keeping up with the very, very fast pre-printing that was happening in the COVID-19 literature space. So all of those were indexed so that people could search them via this portal. Talking about the structural biology, there's now a large number of structures in the PDB. EMBL colleagues in Heidelberg came up with one of the first EM views of the virus, and you see that there's this flexible linker, so the spike protein flexes around. That flexibility looks like it's important for the infection cycle of the virus. In Grenoble, we looked at the structural biology of the polymerase, and it's still a credible drug target to see if you can get into the polymerase. It would be a good thing to have more antiviral drugs that we could use. And that's also the case for the protease as well, of looking for drugs, in this case, on the protease. This was the data platform, the COVID-19 data platform. And one of the things we developed here, there's an interesting dynamic between the public health agencies and fully open research. And public health agencies are often very happy to share their data, but they want to be in control, and in particular, they want to be in a situation where they can inform their citizens and their governments about what is happening in their country before the data goes further out and informs the world. So that level of control is tremendously important for public health agencies and governments, unsurprisingly. And we came up with a system called data hubs, which allows, in this case, for example, Denmark, to have a data hub where the people in charge of the data hub can specify when they publish and make the data fully open for other researchers to use. And that gave a lot of trust to the public health agencies that they were in control, and ultimately allowed more data to become open faster because we had given control to the public health agencies for this management. But I'm going to now talk about a piece of research that's done by my colleague Moritz Gersten here. And I'm going to start with just a video. Now, what you're going to see here, this is England, which has been one of the best studied locations genomically for SARS CoV-2. So the genomics present in England was pivoted to tracking SARS CoV-2, and so we have a remarkably detailed view of SARS CoV-2. I'm going to show you the video here and this is very early on, and it was before WHO had given these designations alpha, beta, gamma, delta. So this is called B.1.1.7. We now know this as the alpha variant. And what you're going to see here is a deconstruction of a period across of the pandemic in England, and you'll see the rise of alpha and the removal of many other regions of other variants across the UK. So here we go. Hopefully this works. So there's an outbreak that happens in the autumn in Manchester. This is from the kind of Wuhan-style variant. It actually comes via Spain, I'm afraid. And it's going to fade away. Now watch down here. You'll see something turn up down here. This is the alpha variant. It goes hot, and now it's just going to go across all of England. So being able to deconstruct the infections is non-trivial. And behind that animation is quite a sophisticated model, which is taking the results of RT-PCR testing, and this is just showing Cambridge here, and also the genomic surveillance data for different variants, and knowing how infections happen. So infections, one person passes the infection on to other people. There's a gap of between 5 days in the case of the alpha variant, and so you can fit how you detect things after people become symptomatic. So you don't detect them at the point of infection, you detect them 7 to 10 days after the infection. And then, of course, this data about the surveillance comes later. So Moritz was able to combine all of these pieces of information into one model, so in particular the sequence and the RT-PCR data. And that allowed him to produce the sort of animations shown here in now a sort of simple form of different months over this. And this is one particular, this is averaging the behavior across all over England. And here is the rise of alpha going up here. And this is the alpha line here. And you can see that alpha turns up down in the south of England first, spreads, and basically outcompetes all these other lineages which were circulating at the time. And there's very detailed geographic inference. And one of the things, geographic and time-based inference, so I'm just showing you different parts here of England and how this is split out. This plot here was a completely key plot because because Moritz, there was, if you're not English, you wouldn't know, in November of 2021, there was a mini lockdown, circuit breaker, it was called. 2 weeks over half term, everybody in England was asked to leave. And at that point, alpha was present in Kent and the southeast of England, but not present elsewhere in England. And in fact, total incidence across England dropped at that time. It was sort of down, this little bump down here in these places. But importantly, Moritz was able to show that the alpha was going to transmit even through that lockdown and even when schools were closed because of this circuit breaker. And that's what this plot shows here, which is the change in the rate in alpha in the second lockdown, the third lockdown and the second lockdown versus other lineages. And the fact that alpha was still transmitting during that lockdown was a key piece of information to realize that the rate of transmission of alpha was far, far higher and would not be held by without shutting the schools. And this is another output of this model, which is the relative growth rate, where one virus is arbitrarily chosen, B1.1, as sort of one. This is the original A and B lineages that came from Wuhan. The A lineage died out. The B lineage is the lineage that led to everything else on the planet. And you can see here the relative rate bounces around in these different lineages till you get to B117, which is this about 60% faster transmission of B117 versus everything else. Now, it was a really fascinating time. I don't know if you can remember that time back. It was Christmas of 2020, the New Year of 2021. And if you remember, Alpha was the first serious variant that changed the response that we had to make in the pandemic. And so we presented the information here to the UK Prime Minister and Cabinet, and so it's part of the science package that went into that things. We didn't present it directly to them, but we did provide the science information. My colleague Rolf did present it to Angela Merkel, which I was slightly jealous about, but he is the German-speaking colleague, so he presented it to the German Chancellery, and it was part of the science advice then. And I talked to my colleagues in France who were informing Macron, and again, my French isn't good enough for sure, and I'm also too junior, but we were part of the science package that informed the decisions that Macron made in January. We also shared it to all the member states via delegates and leading scientific colleagues, and in that context, it means here, Alfonso Valencia got an urgent message from me. I said, Alfonso, you've really got to look at this and persuade anybody to look at it. And we were right. Now, it's quite interesting. At the time, there were a group of people who really didn't want there to be a faster transmitting variant. Many French people were convinced that Boris Johnson was just crap, and this was an excuse for the Brits being awful at pandemic response. So we had to say the Brits are probably not very good at pandemic response. We agree with that, but that doesn't mean we made this up. This alpha variant is real. And these are some of the other bits of supporting evidence that went into this. So it has a higher viral load. This is this key business that it grew from low levels of September onwards, including the November lockdown. And then this is a piece from colleagues in the Public Health England, HSA as it's now called, doing contact tracing and showing that the alpha variant showed higher forward transmission from one infected individual, more individuals, by the contact tracing approach, was shown to be infected. So it was a real tense moment. Moritz worked his guts out to put this model together over Christmas, crunch the data, and do it. Utterly amazingly, we had to do it all over again for Delta. Now, I don't know if you remember Delta. That was the variant that almost certainly originated in India, expanded over India, and then started to appear in Europe, in particular in northwest UK was one location for this. So here's just the same plots. It's now got a new line, which is, there's B117, this is alpha. And then here is this B1617.2, now called Delta, rising up here. And that's it coming up suddenly and outcompeting alpha. And there it again, coming up and outcompeting alpha. So the second time around we did this, it was sort of really interesting. Again, there's a lot of debate at the time about what are you seeing in your data, and is this really a new variant, or is this just a lot of people arriving from India And there's a very careful piece of modeling here about showing that the rate is really higher for delta versus alpha. And again, that call ended up being correct. But we had to go through the whole rigmarole again of persuading people that we understood the data correctly and presenting it again to colleagues across the world and across Europe. So what I've shown you here is something which I've really been part of and I should have added a piece of work that we're doing on contact tracing, where we're using the genomics of the virus to understand which part of the contact tracing procedures have worked. So the interesting thing about the virus is about every third infection, the virus sequence will mutate and be passed on. So the mutation will be passed on. So there are about 10,000 bases in the virus. Most of those bases you can change, and it doesn't change the virus that much. Every third infection, you get a slightly different virus. So you can build these 1 million viral phylogenetic trees, or I think it's about 500,000 for the UK. So 500,000 viruses, and we can infer the true phylogenetic tree of that virus. And from that, we can work out which bits of the contact tracing worked or didn't work inside of the UK, and where infection really happens. So a slightly unsurprising thing is that if the hairdresser is infected, they transmit to pretty much all their customers. And so you get these little bursts around hairdressers. They're rare, so hairdressers don't get infected that often, but if they are infected, they really infect a large number of people afterwards. Another thing that we could show there is that shopping, people in the same shop at the same time tend not to infect each other, which is a useful thing for policymakers to know, which is, you know, on average, shopping is safe for transmission, whereas you can see that maybe you would want to pass out lateral flow tests to hairdressers if we go through this again. But that's the kind of level of detail that you can get into using this data. So this has been an amazing 2 years for me because it's really opened my eyes to how healthcare operations and research interact. And I'll come on to another place where we do this. But this is, there are some very differences here between healthcare. So healthcare is very data-driven these days. Most systems are very computational databases. You have to have, you know, check-ins and there's devices that measure things on people, and they're all got computers everywhere. Everything has to happen fast in healthcare. Doctors have to make decisions very quickly. So there has to be an incredibly fast turnaround of this system on the left-hand side. You have to provide robust analysis, for example, in this pandemic situation where we're calling whether there's a new variant. Those decisions have to be made often within days, maximum a week. And so everything's got to work really, really well. You don't have the ability to come up with radically new method. So you've got to use methods that you trust and that you can run very quickly. But at the same time, you need to work out how you integrate new knowledge and new research into this loop, because there are often questions that you would like answered, and somebody's got to come up with the ways of doing that. But this loop ultimately provides data, and I've shown some of the analysis that we can do with that data. And then there's this kind of research component, which is much less fast. There's a business of like understanding how the virus is really transmitting. I'll come on to another example about human biology in a moment. And these, a couple of weeks or a couple of months to come up with a new analysis system is quite sensible, actually. So it's a much different rate of times. There's this exploratory open-ended analysis. When you're in research mode, you don't necessarily know precisely the question that you're going to answer. I know so it really freaks out the policy people. You're like, can you just give me the data? We're going to see if we can understand it a bit better. And that they don't, the other side doesn't like to hear that all the time. One really interesting thing is the process of understanding your data better often leads to very deep QC, and that goes to this business of these, the importance of raw sequence deposition for the virus. Without raw sequence deposition, we would have thought there would be many other dangerous variants knocking around, because all sorts of weird and wonderful errors occurred in the processing of the raw sequence to the final result in certain settings in certain places. Very, very subtle artifacts. So not obvious things. It's not that people are being stupid or difficult or they're not getting it right. It's just very, very complicated to do these things. And so this deep QC is a real feature of what researchers do when they're presented with these data systems. And of course, ultimately, you create these new analysis schemes. I showed you one there by Moritz about following the progression of being able to put together genomic sequence and RT-PCR testing into the same framework to analyze the relative growth of these two things. And that is the thing that you want to need to push over there. And it's not just the pandemic. There are many, many settings in healthcare which have this same feature. And this infinity loop it also features in Genomics England. In fact, the name comes from Chris Wigley, the CEO there, about emphasizing that we have two loops in Genomics England, a loop which is about research delivery and a loop which is about operational healthcare. So the operational healthcare is generating human genomes for the diagnosis of rare disease and cancer. I am very proud that the NHS in the UK will now sequence the whole genome of children with suspected diseases and their parents as a routine part of the NHS delivery in the UK. So if a child has a suspected rare disease of one of a number of different classes, then the clinician can say, I'd like to have this child and their parents sequenced, and they go into this loop here. It is very effective, by the way, in diagnosing rare disease of children, so it really does work extremely well. But these data sets become the data set for researchers. Again, a really interesting thing is spotting complex errors. You know, we don't, again, not anything that, nothing stupid. These data sets are very, very complicated, and so you get kind of really complicated errors that can kind of come through. But in this case, discovering new disease genes, discovering new variants in disease genes, and therefore producing new ability to do diagnoses back in this loop over here. And this is going to be repeated, I hope, for cancer. That's going pretty well as well. And as many of you know, Arkady knows, Roderick knows, I have a passion for, frankly, everybody on the planet to have the benefits of this, but in particular, all the citizens of EMBL member states, Spain included, Switzerland included, France included. So I have a passion of trying to make this technology come into the healthcare systems across Europe. And in the new indicative scheme, our new scientific program of EMBL, we have the genomic medicine platform, which is kind of fancy name for these 5 different things. So one is strategic advice. It's almost literally having a Zoom call with Ewan Birney and colleagues, if people want it. The second one is the reference data. I mentioned that the EBI provides the reference human genome and the reference annotation. I think tomorrow or on Thursday, there will be the release of the main project, an agreed set of transcripts between EBI and NCBI, which are to the base pair identical between these two sets. Now, only bioinformaticians will get excited about this. Everybody else either says, what is a transcript? And that's one question. Or if they know what a transcript is, they'll say, surely you've done that already. And then you're like, no, let me tell you how painful it is. And these really edge cases and these really weird things that happen in the human genome. So it's that kind of attention to detail for reference data, which is really, really critical. And then here, with colleagues here at CRG, the EGA Federation, shown with these little spots here, including that lovely spot in Barcelona, for building out a federated system for clinical research and potentially useful for clinical practice as well. Technical and training courses, and then a fifth thing is mid and long-term secondments of people to EMBL to upskill to gain more skills in this area. So with that, I'll end with thank you for listening. Thank you to my EMBL colleagues that I've mentioned, in particular Maritz Gerstung and Samir Velenka, and also just to say that the goal here is to work for Europe and the world in delivering not only great science, but delivering the benefits of science to the citizens of the countries we live in. So thank you very much.
So I think we do have time for a few questions. I have many, but I'll let you start. We should shout a bit. I know you don't. Should I shout?
Yeah,
you don't need to. That's fine. It's all convenient.
Okay. Yeah, thank you. It was a very nice presentation. Thank you very much. So I have a couple of questions. It would be like, for instance, you have mentioned that this has been like an amazing amount of sequencing to monitor the pandemics, but what would be your recommendations for a country that do not have the power to invest in such deep sequencing of so many samples? So what would be like the key things that one should target?
I mean, actually, I think it's a question that's asked to us and to other colleagues. The key thing that people sort of settled on is at least 5%, if not 10%, during the pandemic of your RT-PCR samples, positive samples being sequenced. Looks like a good place to start. And then the other really subtle thing is trying to make that sampling as random as possible. And that is actually quite hard, because the easy thing is to do, you sequence the sequencer is in a particular place, and they're really close to this other place that does the testing. So logistically, it's easier to just like connect those two things up, but then you're only really sampling that testing site or that region. So it's sort of 10% random, or 5% targeted, 5% random would it's not a bad place to be. It is interesting. The UK has just sort of switched off a lot of its sequencing, and they're going just back to random sampling. Something the UK did, which was quite sensible, is they random sample. They don't just use symptomatic tests. They random sample people. So they're now relying on random sampling a lot more to understand this. And whether it was wise to step away from the sequencing now, I don't know. But certainly the random sampling has been really, really important. And we should do a lot more random sampling, probably routinely. But also, if another pandemic starts, you've got to hit the random sampling button very, very early.
And then my other question, and I will let others to ask questions as well, would be if you, sorry, now I forgot the question. Yes, now it come back. So the question would be, what are the things that the EBI, that were advising policymakers and all this, what would you do differently now? What are the things that you have learned that if you had done it different, they would have listened to you better or something?
Yeah, I think I have a sort of personal take on this, but it's important to realize where expertise like myself starts and ends. So I'm not an expert on public health delivery, for example, and I'm not an expert on infectious epidemiology, though I now have hung out with infectious epidemiologists a fair bit, so I do know a lot more. I mean, there's a real meta thing. I don't think we, in Europe, I don't think we really appreciated the importance of the fast response and the importance of good operational public health shown by the East Asian companies, so in particular South Korea and Japan. And if you look at what they did very, very early on, and they continue to do, it was very, very quick, and it was subtly different from how we did it in Europe, perhaps with the exception of Germany. So, so I think there's something to learn there, and I'm trying to bring the genomic data alongside of our contact tracing data, now alongside the UK's contact tracing data, to show that in more detail. But it's an important thing to realise that there were countries that had a very fast and good response, and it's worth us learning from those countries. And I think that all the conversations tended to be national. All the science and the understanding tends to be international. And that is a kind of another thing to emphasize, that viruses absolutely do not care where you live and what passport you have. Absolutely don't care. They just care about infecting humans or these sorts of things. And so there is a lot of importance of getting the international discussion, coordination, not just of data, but also of best practice and knowledge done.
Thank you very much.
Thank you, Claudia. Thank you. We do have time for more questions, but I cannot avoid making a statement, just using the last things you say. Essentially, one thing that has been interesting in the pandemics is that we all here are aware that science is not the project of a group of researchers or a given PhD. It's not the project of an institution. It's not a project of a city or a country. It's a project of the whole of humanity, right? And bringing this realization to politics that is dominated by states is kind of very complex. But if we want to press the random sampling, data sharing, good practices sharing button the next time, we'd better bring these practices to the general policy making. Anyway, this is not my talk, it's his. So more questions for him. Anyone? There's two. Steve here. Well, the mic is closer there. I'm afraid I don't know your name.
Sylvia.
Sylvia, please, go ahead.
Following what you said, that maybe scientific conversations are global, while maybe projects and resources are provided more in a national manner. How do you think in the current moment that we have so many genomes to analyze, so much data to analyze, how computational resources should be invested? Because sometimes they are done from a center point of view, that each center have their clusters, but maybe if we need to analyze so much data like now, how we need to organize our resources?
Yeah. The most important thing is that it works. I know that's obvious, but there are many ways, I think, of doing it, and they range from smaller groupings of computational infrastructures in departments, sometimes labs, sometimes, and then they go up into institutional things, and then they go up to national things, and then they go up to public clouds, commercial public clouds, where people buy access time on commercial public clouds. Pretty much all of those are valid. The smaller it is, the harder it is to cycle your capital and get good infrastructure provision. But of course, then priorities are the bigger it is, the more sets of competing priorities there are around it. An important thing to realize is that although we can share bandwidth, for example, so bandwidth network if you don't use, if somebody does not transmit data this second on this part of the network, you can never get it back. So there's no, although when the network is congested, you have to think about sharing the network. When it's not congested, you don't have to worry about it. And that is one of the beauties of the Ethernet protocol. If you go back to the 1970s about what the Ethernet protocol does, it really doesn't try to manage congestion or fair sharing. It just says, if the network's open and you can send, just go for it. Now, this is very different from the disk. So disk is very, very different. If I store 4 petabytes of astronomy data, I cannot reuse those 4 petabytes to store biological data. I have to decide what I'm storing on disk. So these are 2 computational resources, network bandwidth and disk. And from the outside, you might think they behave very similarly. But in fact, they don't. They don't behave similarly because of the kind of constraints around them. So I think when you design these systems, you have to, people I think sometimes make inappropriate mistakes. And very broadly, network is best done centrally, globally, nationally, that sort of thing. Disk has to be tied to your projects, because if I store data, no matter where I store it, I can only store that project. Compute is kind of interesting because it sort of sits in the middle. Because the compute needs the data, you've got to arrange things correctly, there's got to be some other things, but if I don't use my chip this second, it's a bit like the network, then I've just not used my chip this second. And if somebody else could use it, then maybe it should be fine to use it. So compute is actually the most complicated one to arrange compared to network and disk.
That was a sophisticated answer, but very useful one too, when one has to design collaborative projects. There's another question here by Steve.
Hi, Ewan. Thanks very much for the talk. Just a quick question. So given that we're about 2 years into the pandemic so far, do you feel that the pandemic has accelerated biomedical science, or has it hampered biomedical science as a whole?
I don't know. I don't think there's an easy one. I think it's changed it a lot, I think. And actually, a lot of my colleagues who are closer to public health and infectious epidemiology, infectious biology, they're very, very worried about complacency setting in very quickly. And if, these people have been saying for the last 20 years, look, if one of these viruses goes global, we are in deep, deep trouble. And sure enough, one of these viruses went global and we were in deep, deep trouble. And now they are worried about everybody just like, oh, that was awful. Can I just forget about it, please? By policymakers and citizens, I think. So I do think we need to think carefully about we're not out of the pandemic, and as I'm sure you all know, this virus is too transmissible. This was clear April 2020. It is too transmissible now for us to contain. It's also in a number of animal reservoirs. So that's it. We are going to have the 5th endemic human coronavirus will be SARS CoV2. And it's definitely, definitely nastier than the other 4 coronaviruses. So there are 4 circulating coronaviruses. We call them the common cold often. And they do give a disease in the older age called viral pneumonia. It's not perfectly the same as COVID, but it's not, there's definitely some lung-based clotting, some other things like that. But there's no doubt that this virus triggers a far worse disease in many, many different human settings. So obviously the ones that we concern us are the ones where you end up in hospital and then that often go to ICUs and often die. So that pathway is very bad. But actually the long COVID pathway, which is for younger people, often women, so that older is older and often men. It is kind of interesting. The long COVID is younger and often women. This is close. It's really not nice at all. So we've really got to have a plan for the next, for the foreseeable future about how we manage just this virus, let alone another pandemic. So has it improved? Has it helped science? I think it's placed, the economic impact has been gobsmacking. So my colleagues who I know in economics, they are like, this is ridiculous. This is, it really was warlike levels of economic destruction. And it should be the case that society tries to mitigate and insure itself from ever having to do that again. We should really know, we should be much better at these things. But again, this complacency, people worry a lot about the complacency. They worry about other priorities coming in to that. So even though rationally you'd say, look, okay, sod it, let's just like quadruple the science budget, PS, infectious disease, we really don't like it, let's make sure we don't do that again. That could be a rational response. But funnily enough, we have to almost make the same arguments that we had to make before the pandemic yet again about this. It's like somebody else is going to look after it. It's not going to happen in my period. Could we just forget about it? So I do worry. I do worry about that for sure.
That's exactly like that. There was this phase at the beginning of the pandemic when we thought, hey, we are going to come up as better people. Then we thought, well, not better people, but at least we'll have larger science budgets. And now we think, oh no, come on, we are back where we started. Yeah. But anyway, I think this was a great talk and a good bunch of questions and great answers. So with that, I think we are out of time. So we should probably thank you and all of you again, and see you next time.